Have you ever wondered how our nervous system, like our neurons in our brain, can react so quickly? Well, the reason is because of ligand-gated ion channels.
Ligand-gated ion channels are transmembrane ion channels that open or close in response to the binding of a chemical messenger like a ligand. A very common place to find ligand-gated ion channels are in electrically excitable cells like neurons. The reason why is because these ion channels react quickly to the binding of ligands, so they're very commonly found in cells that need to react quickly to stimulus.
Let's say we have an ion channel that looks something like this. This particular channel at the moment is closed. Right now we have our intercellular environment. This is where our cytosol and all of that stuff is. Notice the ligand approach the channel. Only specific ligands can bind to specific channels. This is what we call an induced fit. Once this ligand binds, it'll cause this closed channel to open up.
Now this is our open channel. Once the ligand binds, it can control the opening and closing of the ion channel by altering the protein conformation of the entire protein. A channel opens in a different place and the ion permeability of the entire plasma membrane can quickly change. Remember, this is not just one thing happening. When these ligands are binding, there are many of these channels scattered throughout the cell membrane, so all of these are opening and closing at once.
Once this channel opens, it'll let ions move through the open channel. Once these ions are moving in and out, this will cause a change in the electrical properties of the cell. This extracellular ligand signal converts into an intracellular electrical signal. Once these ions move in or out, you'll have an intracellular electrical response, an electrical signal happening inside the cell.
In summary, ligand-gated ion channels are one type of membrane receptors. They are transmembrane ion channels that open or close in response to the binding of a chemical signal like a ligand. Once this ligand binds to an allosteric site, which is a site that's not on the channel itself, it'll let ions such as potassium and sodium and so on move through the membrane.